Sean,
Okay,
did you give me the go ahead to start?
Yes, I did. Yeah.
Okay, all right. So this is the last section, so this is not going to take too long. We have maybe 5-6 slides between what I'll present here right now, and then I'll give the word to Andy. So this is related to the last and fifth research area, that is genetics and genomics. The first request was request 18 from the SRB to provide information on the annotation of the genome. And at that time, at the time of SRB017, there was a request to NCBI to annotate the genome, and now we can say that the genome has been fully annotated by them, and using all the RNA-seq information that we have generated at the IPHC so far. In this little box here, you can see some of the metrics of the genome, close to 600 million base pairs, 24 chromosomes, 20,000 genes at a 91% coverage. And the annotation, sorry, no, it's not fast. So the annotation now contains, the specific positions of the genes within the genome, and importantly, also information on the actual identity of the genes that are present in the genome. So there's a web browser that contains all that information, that it's housed at NCBI, and it's public. It's of public access. The next request
before moving on, I guess great to see that you're continuing to move forward with some of the bioinformatic steps. I guess maybe the SRB wasn't quite complete in its request. I guess another request, and maybe you could comment on this now, would be, so what do you do with the annotation? You've got a genome, you've got SNPs, you're going to have whole genomic data, you have transcriptome and proteomics data. How are you going to use the annotation?
Well, the annotation is key to identifying, the composition of the genome in terms of the expressed genes. So any study that we do from now on that maps against the genome will contain information on what genes are present there. So, for instance, if we start with the transcriptomic data, any new RNA-seq study that we embark on, now we can, in fact, map all the transcript reads onto the genome, and then identify those potentially differential transcripts with the gene annotation associated with them, so we can know exactly which genes are being regulated under different conditions. But all the population genomic studies that we're going to be doing, and that Andy Jasonovich will speak to in just a minute, we'll definitely use the annotation of the genome. Any further studies that we do down the line regarding hopefully landscape genetics or genome-wide association studies, or any studies in which we identify potential markers under selection, and we need to identify whether those markers are potentially of functional significance, this annotation will be key to provide that information. So I think it's quite well understood that a fully annotated genome is an incredible resource for down the line applications related to genomics and population genomics and transcriptomics. For the sake of time, if you allow me to, I'll continue. Were there any other comments related or questions related to this request?
Doesn't seem like we do, so continue.
No? Okay, if not, then I'll move on to the next request, and then I'll pass the word to Andy Jasonovich, who's going to be talking about this next few slides. So Andy, go ahead, please.
All right. Hi. Can everyone can hear me?
Yes.
All right. So yeah. So the next couple of slides here are in response to the request that the SRB made. That the IPHC Secretariat include individuals of different ages and locations in the Gulf of Alaska and Bering Sea in their whole genome sequencing analysis, including individuals from different places in the Gulf of Alaska and Bering Sea. And so, yeah, I just wanted to provide kind of a summary of the samples that we actually have available to, address this, research objective. Oh, next slide, please. All right, so I've prepared a couple of maps showing the locations of the tissue samples that we have available for genetic analysis. And so the top row of maps shows the sampling locations of individuals for which we have a tissue sample and we know the ages of. And then for which we know the ages of. So, and then the bottom row of maps are the locations of fish which were collected on the NIMS bottom trawl survey. And those fish were fin clipped and then tagged and then released. So we do not know the ages of those fish. However, we do have length data associated with them. And we have far more samples that were tagged and released without any ages than we do age samples. Next slide. So I've put together these tables to give you an idea of the exact numbers that we have in each of these different groups. And so I've broken it out by area and then year, and then for the age samples, area, sampling year, and then each individual age up to age class up to age 5. And so initially we had proposed to use length age key to indirectly assign ages to the tagged samples so that we could utilize them for this to look at genetic diversity among juvenile Pacific halibut. However, after some discussion with Lori and Ian about the use of those keys, they may not necessarily be provide us ages that we can use to select samples with, due to the fact that there's overlap in the length distributions among age classes, and then and then they would be assigning ages in a probabilistic sense, and that overlap is would potentially cause issues there. It wouldn't necessarily generate very clean age assignments, and those keys vary year to year, and then among the different areas as well. And so I kind of see two different ways about selecting samples for this analysis. Focusing our efforts on, say, the aged samples for which we know the ages of, and that would restrict it to, a relatively small area for which we have collected samples from in the Bering Sea. And then, you can make a comparison in 2009, between samples collected in the Gulf of Alaska, or focus on, the tag samples and, say select for age ranges, for instance, select a size range that would correspond to a likely age range that would indicate that those are juvenile fish we're looking at, and maybe compare between 2017 2019. And so we're thinking about that, and if we discussed this a little bit with Kim yesterday, but if any other members of the SRB have any input, that would be great to hear. So next slide. And so, yeah, so really the overarching goal of this, research is really to infer the potential contributions of fish spawn in different areas to the Gulf of Alaska and Bering Sea as kind of a complement to Lori and Ray's larval advection modeling work. And really, that's going to come down to comparing genetic diversity metrics between those two areas, or sub areas within the Gulf of Alaska and Bering Sea. Testing for deviations from Hardy-Weinberg equilibrium, and then estimation of admixture proportions, so we can infer the number of different genetic groups that might be using these nearshore areas. And so, yeah, I think with that, yeah, so if there's any comments or anything, we could take those now before we go on to the next request. All right.
Sounds good. Sounds like you can move on.
Move on to the next slide.
Yep.
All right. So the next request, request 15, is that the SRB requested specific information on how the sequence data would be mapped to the reference genome. And so for this, we're going to use methods that have been applied to similar studies, or studies that have used also used low coverage genome sequencing. In particular, Klukas et al. 2019 applied very similar methods to conduct a similar analysis with Atlantic cod. And so next slide. And so this slide here essentially outlines the workflow that we're going to follow. So each step is a description of the step and then the software that we plan to use. So first off, the first step is to actually align the raw reads directly against the reference genome. The next step, we'll remove any PCR duplicates. So during the library construction process, there's a PCR step, and you want to bioinformatically remove any reads that are artifacts of that, because that can bias the genotype calls that you get, or that bias your genotype calls. Next, we'll filter low quality alignments, and this could include alignments that might possibly align to multiple locations in the genome. We'll clip overlapping read pairs. So the data you get back is they're paired sequence reads, and in some cases they overlap, and in those regions where they overlap, you want to account for that so it doesn't affect the depth at which you can call those called genotypes. And then finally, we'll perform indel realignment to essentially improve alignments around insertion deletion elements, using all the, essentially using information from other sequence reads at those locations in the genome. And at that point, we'll have what you could consider as analysis ready alignments, or sequence alignments. And so next slide. So I put together this slide that, just kind of reminds you of the different analyses that we proposed and plan to do with this data, or the downstream analyses we plan to do. And so really the first step will be SNP detection and genotyping, and then from there we'll use those genotypes to conduct these various analyses. And I've broken them out into 3 different groups: estimation of genetic diversity metrics, analyses of population structure, and then genomic analyses where we'll actually try to detect outlier loci or SNPs that might be subject to directional selection. And so, this is one area where it'll be really great to actually have that annotated genome, because we can, then compare, the positions of these SNPs to say, the function of certain genes in the genome, for instance. All right, next slide. All right, so quick update on where we are at with this. So earlier this month, we submitted a trial library for sequencing, and that library contains 36 samples, and it's being sequenced on an Illumina HiSeq 4000. And really the primary objectives for this trial library are to validate our library construction methods. So initially we had planned to utilize lab equipment and lab space at the Northwest Center to construct these libraries, but due to COVID-19, you know, accessing that space is not possible at the moment. And so this will be a test of our modifications that we did to those protocols, so we can actually construct these libraries at IPHC. We'll also be assessing the genomic coverage of the individual samples in that library, which you know, essentially influences how many samples you can pool together in a given sequencing run. And which is essentially a function of the genome size and the output of the sequencer, because, you know, for further sequencing efforts, we hope to scale up to a larger machine, or a machine that has larger, more sequencing output. And we just want to make sure that math is correct using this run. We'll also be able to evaluate the accuracy of those genotypes that we call, because 30 of those 36 samples were used in the study that was used to identify the sex-linked SNPs that are used for the genotyping of the commercial catch. And that data is RADseq data, which is you know, higher genomic coverage. And so when you have a greater coverage at a given genomic location, you can call SNPs with more confidence. And so those genotypes, you know, we have very high confidence in those genotypes, and so we can compare our lower coverage data to that in order to validate that. And then finally, you know, test some of the software that we want to use and gain familiarity with that. So next slide. All right, and so yeah, on this slide, this is a map of the different sampling locations, sample collections, the locations and sample collections we have available for our research on population genetic structure, which, you know, feeds into that analysis of structure and regulatory 4B, area 4B. And so the years highlighted in green, or the years highlighted in green are sample collections that were included in this initial sequencing effort. So, you know, this is really kind of the start of data generation for this project. And I think, yeah, that's it for this. If we have any questions or comments.
How are we doing for questions or comments on this?
All right, just it's not really a question, but as somebody who is not that familiar with the processes, particularly of the software, I still find it very useful to have an understanding of how these things go together. So I just wanted to say that I think this is more of the information that will allow us to make some of those decisions at least, and evaluate the efficacy of some of these research projects. So I just wanted to comment on that.
Sure, thanks.
I think in this area, and we talked about this before the primary meeting in a separate meeting yesterday morning, that the narrative provided in the document to the SRB was rather general, missing information, and you know, talking through things with you know, Andy, Joseph, and the group. You know, there was a lot more meat on the bones than what was provided, and again, you know, sort of a recurring theme here is that for us to be able to evaluate what is being proposed, you know, having some more meat on the bones in terms of methodology would be really important.
Sure, yeah, absolutely.
Okay, thank you. Thank you, Andy.
Yep.
All right, so this is Giuseppe again. This brings our presentation to an end then. Thank you.
Okay. Great. Before we move on, did anyone want to add any comments, questions, clarifications on the biological and ecosystem research program?
I had one comment or question going back to migration. And maybe this is, you know, a fruitful topic as the Secretariat is considering what areas might be useful to move into, you know, for the next 5-year plan. You know, I'm not that familiar with oceanic dispersal of gametes and larval fishes, but what information I am aware with is that, you know, a lot of the gene flow, a lot of the movements occurred prior to settlement, and that's at the fertilized egg and very, very young larval phase. So the work that Lori, Ray, I mean, the whole group that has done on larvae and juveniles is really, really useful, really, really interesting. But I would offer that much of the research has been done on pelagically dispersed earlier larval phases, and that's not to suggest the Commission can get your hands on these things, but in many ways you don't have to, because a lot of people in NIMS and other areas have been using things like particle tracking. So work that the biological group has done has identified areas and depths where there are known spawning areas and known times of year. And so you could use things like particle tracking released at certain times in certain areas at certain depths, and you could recover that and get an idea of where really most of the action is probably at in terms of mixing, you know, during these pelagically dispersed phases. And I was just wondering if there has been any thought to moving that forward as another research area in the future for this next 5-year plan.
Yes, if I may add, that is precisely what was done in that paper that was just accepted for publication in Fisheries Oceanography. There was that modeling effort was based on tracked particles, so it was actually not relying on actual larval collections, but as a result of the modeling of particles that were tracked and released virtually from a set of predetermined spawning locations, 5 in total.
Okay, I guess I need to read the paper.
I will send the accepted version of the paper, which was slightly modified from the one that you received in June, and for the SRB to review.
Okay, thank you.
Joseph, could I offer a comment question at a slightly higher level about the presentations in the biological session? I find these incredibly interesting and useful, but and maybe this is partly because I'm new to the SRB, I sometimes struggle with figuring out where comments and advice are useful and where you've really gotten that part nailed down, and it wouldn't be helpful at this stage to offer advice. So I wonder if going forward, it would be possible for the presentations to just highlight areas of current uncertainty, places where you have some decisions to make in the research, where the SRB could weigh in a way that would be helpful to you. You know, I want to make sure that my comments are not coming too late to be useful or focusing on something that you've already identified as clearly established. Does that make sense?
Yes, yes, and thank you for the comment, and the point is well taken. Thank you.
Thank you.
Okay, sounds like that completes the biological and ecosystem research section. Thank you very much to everyone who presented, along with Josep there. Very informative talks, as you can tell. Everybody sounds enthusiastic about the research that's happening. So I think now we can make a transition to the MSE. And I'll just let Alan set up and take it away when you're ready.
All right, thanks, Sean. If I could be given control or share, there we go. Excellent. Just to confirm, can everyone hear me and can you see my screens? It has a PowerPoint presentation up.
Yes.
Yes, thanks.
Excellent. Okay, feel free to interrupt at any time in this presentation, and I look forward to having this discussion with you. So this is a presentation of the management strategy evaluation, where we're currently at, and I'll be showing you results in a moment. And just to note that, as you already know, we do have an MSE team, what we call the team, with myself, Pierre Carpi, and Steve Barikoff are part of the main team, and then Ian Stewart has a lot of interaction with us on the specifics of the MSE. I also wanted to note that the we're in full production mode, and the MSE is being updated by the second, actually. And in fact, 30 days ago when we released the document is a long time in our MSE world. So a lot has been done in that last 30 days. We did not submit a revised document, but I notified SRB members maybe a week or so ago that we had results available in the online tool that Dr. Branch mentioned earlier this morning called the MSE Explorer. And I'll walk you through that Explorer today. And with that in mind too, just to let the SRB know that the MSAB 16 documents were released or published last Saturday, I believe. And in those documents might even be additional information that the SRB is not aware of. So what I've done is taken some of those new figures for that and then put it into this presentation. So this presentation has a couple new things that's not in the presentation posted online. So as a reminder, the IPHC harvest strategy policy is what we are investigating with the MSE, and that consists of different elements. We talk about the scale or coast-wide element, and then we talk about a distribution element. And really those are the two key elements we're investigating right now with the MSE, and so we'll talk about that a lot. We also have been talking a little bit, and I know it's of interest to the SRB as well as many others, the decision-making step, which occurs after the management procedure is applied. And that is really separate from the management procedure. And what I've been presenting to the commission as well as MSAB members is to realize that we are investigating management procedures which end here, and without knowing exactly how decisions are being made, this can be a little bit of an unknown at the end there, which deviating from that management procedure may have unintended consequences. So again, a little bit of background on this. In the coast-wide scale or the fishing intensity part, we are investigating various SPR values or the level of fishing intensity. And then as a reminder, we operate under a control rule here with an input or procedural SPR that's then modified as stock status gets low, declining at a trigger, and then going to effectively an SPR 100% at the limit, although some fisheries still do occur even would occur at these low levels like bycatch fisheries and unguided recreational subsistence. We're only investigating the single control rule 3020 right now. We investigated other control rules as part of the coast-wide MSE. And we're only looking at one constraint, and where a constraint is the constraint on the annual change in the TCY from year to year, or that mortality limit from year to year. So if the management procedure says increase the TCY by 20%, but we have a constraint of 15%, the TCY would only increase by 15%. And the same on the bottom end, if it declined by more than 20%, it would only decrease by 15%. There still is interested in this constraint we call slow up, fast down, but that's not a priority at this time as defined by the MSAB. So we'll only be looking at a constraint of 15% maximum change. And so the procedure for distributing the TCY, this is the distribution from a coast-wide TCY as determined from the stock assessment to each of the individual regulatory areas. This is really going to be the focus on the MSE this year, is what are appropriate distribution procedures to meet the objectives. So we have this coast-wide total mortality from the stock assessment separated into components. One of those components is called the TCY, which is all mortality except for the U26 bycatch or non-directed fishery mortality. That coast-wide TCY, we could potentially first distribute that to biological regions using various factors like estimated stock distribution, relative fishing intensities, etc. But that's an optional step, and it's not actually what occurs in the current interim management procedure operated by the IPHC. Instead, we go directly from the coast-wide TCY to regulatory areas currently using stock distribution. That's estimated from the FIS data from the output of the space-time model, actually. And then we adjust by relative harvest rates, what we call relative harvest rates, where basically we're harvesting a little bit harder in eastern areas. That would be 2A to 3A, than in the western areas, which would be 3B to 4B. And that's just because there's some research has shown a difference in productivity between those areas, as well as net migration coming from western to eastern areas. And so a little bit higher harvest rates out in the eastern areas. So we're looking at basically this procedure, and the MSAB has defined different management procedures that may or may not incorporate distributing to region, for example, and I'll explain those management procedures later in this presentation. And then finally, as I mentioned before, the decision-making step is something that I make sure that the Commission and the MSAB members are aware of, that there is this step in the harvest strategy policy. And it's something that we are very interested in, as the SRB also mentioned at SRB017, that they are interested in stimulating this step as well. We'll talk about that in a moment. So that's the background on sort of the general harvest strategy policy at IPHC. And what the MSE team has done is put together a simulation framework to evaluate the management procedures. And as you know, that consists of objectives being defined, the closed loop simulation process with operating model management procedure, and then outputting performance metrics, which we use to evaluate the management procedures against their objectives. And now this whole simulation framework is complete. We have a working framework and are currently running simulations as we speak. So just to touch on a few of those elements quickly, and then we'll focus in on the operating model and the results, unless you want to focus in other areas as well. I'm happy to. But a reminder that with the help of the MSAB, the IPHC Commission has approved a set of primary objectives, what we call primary objectives. And these are classified as biological sustainability objectives and fishery objectives, where all of these are listed in either of these links, but they are pretty much standard objectives, not going below certain biomass levels, stability and mortality limits, and then providing for directed fishing yield. So those three main axes that are common in almost all MSEs are present here. To highlight a couple performance metrics that I'll show you with the MSE Explorer tool in a little bit, we have performance metrics related to biological sustainability. And those are the probability of the female spawning biomass being greater than 20% of B0. This is a very common one we've talked about in the past. And then the probability that the female spawning biomass is above defined percentages in each of the 4 different biological regions. And I'll remind you what those biological regions are in a moment. But basically, the first one's getting at the population in general. And these regional ones is getting at there's adequate distribution of the population across the range so that we're not, so to sort of hedge the bets that we aren't reducing the population to really low levels in certain areas. Now, I note these numbers were defined in somewhat of an ad hoc manner. We will revisit these, but right now they're just numbers that are slightly less than the minimum observed percentages from the survey data that have been observed since 1993. The performance metrics related to fishery objectives are one that the probability of the coastwide female spawning biomass is greater than 36% of B0. And you may ask, why is there a spawning biomass objective in the fishery section? And that is because through some work that Dr. Carpy and I did on reference points and looking at MSY and thinking about it in terms of MEY as well as sort of a buffer for MSY. We determined that a spawning biomass about 36% would be a safe target for an MSY or an MEY type reference point. And so the MSAB saw that analysis, which I believe we presented to the SRB in the past, and they thought, well, this would be a great fishery objective because maintaining the spawning biomass at that level would actually provide or optimize the fishery activities. So we've put that as a fishery objective. The annual change in the TCY is basically in a 10-year period. Does the TCY change by greater than 15% in any of those 3 years? And that's a really informative performance metric for the MSAB members in particular, and they really like that one, as opposed to an average annual variability or AAV type performance metric, which we still have because I do like the AAV, and it's a common performance metric used in MSE analysis. It just provides a slightly different view, being an average of the annual variability. It doesn't really show how often you're actually getting big changes. So I think both of these stability performance metrics are useful. And then in relation to the TCY, we have just the median average TCY. But we also have the median percent TCY in each regulatory area. So what percentage of the mortality limit is in each regulatory area? And then as well as the minimums of the TCY and then the percent TCY in each IPHC regulatory area, to give the stakeholders and anyone evaluating these results an idea of where the mortality limits, how they're distributed, and then what are sort of the minimums that would be observed in each of those regulatory areas. So we'll show some actual results with those performance metrics in a moment, but I wanted to talk first about the operating model. And as Dr. Branch noted this morning in his review, the technical details of this operating model are lacking, and we do realize that. We have been updating the technical document a little bit, and that's been provided to you as document 10. Although really our focus in the last, probably 9 months has just been to get this simulation framework together, working and doing the simulation. So that's really been our biggest priority. And we've been trying to put the decisions that we're making, the assumptions that we're making in a lot of the documents for the various meetings. So for example, the MSAB 15 documents and the 16 documents do describe some technical details as well as the SRB documents. But we hope to in the future, that's going to become a priority now that our simulation framework is working. But I want to point out a few things about the operating model and then show you some results of the operating model. And I do note that at SRB017 in June, we came to you with an operating model that had some issues, not only issues in the fitting and trying to get it to work, but had some coding issues and some errors that we had identified. We've now fixed those errors, and we're very comfortable that the operating model is working as expected, and we're happy with the outcomes of it. So we'll show you those in a moment. But basically, this is what we call a multi-region model. And it's modeling the dynamics between these 4 regions in the IPHC convention area. It's not modeling any dynamics outside of the convention area. So it's not modeling potential movement in and out of the United States or the Canadian EEZs, for example. Within each of those regions, you see there's multiple regulatory areas, and we're not modeling movement between those regulatory areas specifically, but we are modeling the fisheries in those regulatory areas using an areas as fleets type approach, and just apportioning biomass or survey into each of the regulatory areas from within each region. So there's some assumptions that we're making there, but it just seemed to us to be a better method than trying to model movement between each regulatory area, especially when something like 2A contains maybe 1 to 3% of the biomass. That's a small amount of biomass to be trying to model movement to, and we felt it's just a more robust way is to make assumptions across the distribution within a region. There's basically the same 5 sectors, fishing sectors, that are modeled in the stock assessment. We're modeling that in the operating model, and that's, you know, partly a matter of convenience, and the data are already formatted for us to use in the operating model because they're formatted for use in the stock assessment. But these are really the 5 sectors that are most important to the stakeholders for these evaluations. And that includes, of course, the directed commercial fishery. The directed commercial discard mortality is modeled as its own sector rather than through some sort of retention curve, and there's a lot of reasons for that. There's the non-directed commercial discard mortality, which is bycatch, and then there's recreational and subsistence. And talking with MSAB members, we thought maybe we could simplify this a little by combining recreational and subsistence, but MSAB members thought that it would be very important to keep those separate, since they are separate concepts, important to different people, and do operate differently in some regulatory areas. So taking these 5 sectors and then splitting them into all the different regulatory areas, that's a resulting 33 fisheries that we're modeling in the operating model. And you see in a couple areas we have combined recreational subsistence after review with the MSAB, and a few other regulatory areas that's remained separate. But it's somewhat of a large number of fisheries, but it's definitely handled well, and I think it's a good set of fisheries for evaluating the results and providing performance metrics for. For now, we're really focused on directed commercial fisheries as well as the recreational fisheries in reporting our performance metrics. And that brings us to movement. Again, we're modeling the movement between the different regions, the 4 different regions shown in that map earlier. And as a reminder, we showed these plots at SRB016. These plots are probabilities of movement or percent moving at age from each of these regions in the panels to any of the 4 different regions. So you see from region 2, staying in region 2, that blue line, and it's saying there's a really high probability of staying in region 2 once they are there across all ages. And so these are from data as well as assumptions, so tagging data and other things like that. And this has been published in many of the stock assessment overview documents for the past, probably 5 or 6 years at least. And so this was our starting point in the operating model. We said, let's start here, but now we're actually trying to model that. We're actually trying to fit to various observations, such as the actual observed distribution from the survey, things like that. And so we're going to tune and change these movement probabilities in the operating model. So I'll show you what the operating model predicts those movement rates to look like in a moment. But first, this is the outcome of the conditioned model. So a couple of things that we were tuning to, and I'm not saying fitting here because we didn't have as rigorous of a procedure as you might do for a stock assessment, but instead we're trying to get an operating model that represents the Pacific halibut population and that we can apply variability to show a wide range of variability. So in the upper left here is the operating model in blue, and that is just the best fitting operating model. And then the pink shows values from the stock assessment, where the light shaded polygon of pink is the uncertainty from the ensemble, the stock assessment ensemble. So going back in time before 1993, it uses only 2 models. Going forward from 1993, it uses 4 models, the long and short time series models. And then the thin lines of each individual actual stock assessment model within the ensemble. And what you can see is the operating model departs from the range of it as well, especially historically, but in the more recent time period, it's within the range of uncertainty from the assessment model and fits the declines pretty well. It matches it pretty well. So we were happy about that.
Alan, can I stop you there for a sec?
Sure.
Yeah. Can I ask whoever has their mic on to make sure you're muted during the presentations? I have some scuba divers going on that are competing for Alan's.
Yeah, and that's not my kids in the background. So
yeah, I pretty much determined it's the scuba divers coming from someone's mic. Okay, continue.
Okay, thanks, Sean. Sounds better already. So down in the bottom left, we didn't fit to any of this, but this is something I just like to output for my own benefit just to see where biomass was. And this is total biomass now. We do have this by spawning biomass. I just hadn't plotted it yet. This is total biomass in each region, where region 5 is 4B. And so you can see region 3 has the most biomass. Region 2 and 4 are similar, although that early time period is when a lot of mortality happened in region 2, so that biomass declined and has subsequently increased again to be very similar to region 4. And then 4B is always known to have a small amount of biomass, or 4B. And this, so this is just making sure that the operating model is congruent, at least with our assumptions about the distribution of the population. So we were happy to see that it is at least similar to what we think the population would normally look like through that time period. But here on the right in these 4 panels is probably one of the most important data sets to us, and that is the proportion of biomass as observed in the survey in each region, where R indicates the region number. And so what we did in the operating model is we modeled the survey with selectivity from the long areas as fleets coastwide model. And we actually tuned it to match the proportion observed that is predicted from the FIS data and output from the space-time model. And we tuned it to match that as best as we could. Now, first of all, we felt this is a really important data set to match in the operating model, because distribution is one of the most important concepts that we're investigating with the MSE right now. And if we don't have the distribution of biomass right, we're not going to have the distribution of the TCUI correct, and then we're not going to have distribution to the different regulatory areas correct, and it's just not going to make sense to the stakeholders. So we really wanted to make sure we could get this as best as possible. So in each of these plots is the dot shows the modeled survey data. That's the prediction from the survey data. And then the triangle is the prediction from the operating model. And you can see there are error bars on the survey data. And we do a pretty good job fitting this, except in the last year, in this 2019 for region 3 and region 2. No matter what I did, I just couldn't fit that trend. It was really interesting. And then in regions 4 and 4B, it fits very well, except in the early time series part, we weren't too concerned about this, especially since there's very little data coming from regions 4 and 4B before about 1998. The survey design was just very different in those regions at that time. And although the space-time model can predict it, we weren't too worried about that. But in recent years, it seems to match, except for that final year. Not sure why. And that brings us to the movement probabilities. I've displayed this slightly different. And actually, let me start on the bottom right here with recruitment distribution. So the parameters in this operating model are movement probabilities at age, which are actually not modeled as individual probabilities at age by a functional form. And we talked about those functional forms at SRB017. But another important concept to distribution is the distribution of recruitment. So where do the fish, when they're recruited to the population at age 0, where do they go? What region do they go to? So the way the operating model works, it pools the spawning biomass into a coast-wide spawning biomass, produces a single recruitment event for that year coast-wide, and then there's parameters which then distribute it to each of the different regions. So you need to estimate 3 of those parameters, and then you get the 4th, of course. And this is the actual best estimates when tuning this model for recruitment to each of the regions, showing about equal recruitment to 3 and 4, small amount to 2, and then an even smaller amount to 4B from the coast-wide concept. And this, we need to do more linkages with the research that the biological team has done, and the recent paper published by Sidoris et al. But at first glance of this, we find that it is consistent with those results, but it would be nice to do some more direct linkages between their research and the OM. It's just a lot of this is happening in parallel at this point in time. The other plots on this page are movement probabilities at age. So we have age in the x-axis and then the probability on the y-axis. What I'm showing in black on each of these plots are the same as the plots I showed earlier, those movement probabilities that come from the tagging data and various observations, as well as a lot of assumptions, especially for the young fish. And that I believe in previous SRB documents, and I don't remember if we reproduced it in the SRB017 document, but I can steer you to how these black dots were actually produced and from the data itself. And then so that was our starting point. And then to tune the model, we had to modify the movement probabilities, and they ended up in this way, where we also discussed at SRB016 some of the issues we were having were movement was constant over the entire time series. So as a first sort of trial or stab at this is we separated movement into 2 periods, low PDO and high PDO periods, which are the same as the periods as they're defined for low and high recruitment. And what we found is they're not too different between the 2 periods, but they can be different. So let's talk about, say, the movement from 2 to 3. The data showed very low movement, and remember each of these axes might be a little different on the vertical scale. But we have what the operating model is predicting is much more movement from region 2 to 3 than the data and those assumptions actually think the movement would actually be. And it's moving at about 30% of the fish are moving from 2 to 3. And also remember, this is movement for fish from 2C to 3A, as well as 2A to 3B, for example. So it's trying to characterize movement over quite a wide geographic range as well. Something to note in this plot too is this upper red circle here at about 0.35. And so I remind you that there really isn't much data for Pacific halibut, even from the survey, for fish that are really less than, say, age 6 or 7. So what it's trying to gain for all these low age fish, it doesn't have a lot of information to really estimate that from. And what I think it might be doing is, because recruitment distribution here is the same probability over all years in this more than 100-year time series, I think it's saying, you know what, recruitment distribution might be a little different to region 2. And we can account for that between low and high PDOs by just making movement from 2 to 3 much higher for just one single age of fish. So I don't think that this is really one of those things that's saying, oh, fish at age 4, whatever, move from 2 to 3. I think it's saying, I just want to redistribute recruitment in some way. And that's the only way it can do it, because I'm fixing movement probabilities between 0 and 3 anyways. So there's always those types of things which occur in assessments as well. What is natural mortality actually trying to do, for example, while movement's trying to do some other things other than just move fish from on a year-to-year basis. So overall, we have a little bit less movement from 4 to 3, but high movement rates when the fish are young. And we have a little bit more movement between 3 and 2 in both directions, especially when the fish are young. And when the fish are old, those movement rates are a bit different as well. All these other movement areas were fixed, they were not estimated, and that's why the dots are purple and not red and blue at this point in time. It's just something we felt we had to do to simplify the operating model. It wasn't adding a lot, especially with such small movement between these areas. So I'll just pause there. I know this is going to be a big discussion at the MSAB. We've already had some questions from stakeholders and managers about movement and what it means to be departing so much from what the data and assumptions, and I'm making sure I'm noting that there are a lot of assumptions in the black points as well. But I'll just pause for a couple of seconds here in case there are any questions on movement or the conditioned operating model. I think, yeah.
Hey, Alan.
Yeah.
Sven here. You know, I really appreciate the explanation that you give for some of these differences, and you know, it's something that we have to accept when we try to produce a somewhat simplified model over reality. That, you know, there's variance that can be pushed around to some other element. But what worries me, and it kind of goes back to some of the other elements, is to me that says you really need to find out what's happening here, and we need better data on migration, and where is that priority in the research element. The other thing is the sort of circularity with the operating model, and what you're trying to achieve, or what Trevor was trying to achieve with it by actually identifying the uncertainty. If that uncertainty, particularly with the impact on the proportion of biomass in the different stocks, is linked to the productivity of substocks or subpopulations, then I don't see that you run the risk of actually saying, well, migration is so great, I don't really need to do this, you know, on an area basis, because sooner or later the things will move between those areas anyway, or that may be what the model thinks it can do, and so there is that circularity. Do you think that in this case that is detrimental to the conclusions about the risks and the ability to evaluate the robustness?
Yeah, thanks, Sven. I think those are both really, really important points. I'll answer your question, the last question that you just asked, since you asked that as a specific question, but yeah, I think it's a valid concern for sure. And I think the operating model and how it's conditioned and the amount of variability and how that variability is introduced, it should be a main concern of any MSC, and specifically of this one. And yes, when we saw these results and when we're investigating these results, we were concerned that we might be assuming too much movement between these areas such that it does just become a well-mixed population that you can fish out an area and it comes right back, which, you know, may not be realistic. So we've addressed that in a couple of ways. One is through uncertainty, and that is something that we feel is an ongoing investigation for us, is how to properly insert the uncertainty in this operating model, as well as different assumptions about that movement. Now, one reason that I believe this operating model, and through initial evaluations, is that the reason that it has to have so much movement from, say, 2 back into 3, is, I'll just back up one slide here, is this concept here, where the mortality coming from region 2 was quite high in the early 1900s, and it's pretty commonly noted. I mean, I think the commission was formed because of this concern that the population in the CPUE was declining in that period of time. Now, the population cannot maintain a biomass in region 2 unless it starts with a really high biomass, like a really large R0, which some of my operating models went there. And then the alternative way it could explain those high mortality rates and constant movement over all these years was to have movement between 3 and 2, so such that the biomass in region 3, which was not as subject to as high a mortality and was a much higher biomass, could supplement back into region 2 to maintain the biomass throughout this period of time from the 20s and the 1940s until it had those mortality rates were lowered and other things happened to increase that biomass to the levels seen here. So it's not that specific movement data that, you know, some people look for to tell you how fish move, and we do have tagging data back to the 1920s. I'm not sure how useful it would be for investigating migration though. But it's telling us that given the observations we've seen with, there still is biomass in region 2, there was a lot of biomass coming out of region 2, that we have to either have a large biomass in that region or we have to have some mixing occurring. And where to explain these distribution patterns in the later years, it couldn't have those high R0s, and instead it had to have the high movement between the regions. And that's where the operating model ended up, given the data that, or the data that we're actually tuning to. So given that, I think there was the discussion in the biological presentation earlier, the research presentation, about priorities. And I think for the MSE is that migration, movement probabilities, and recruitment distribution is probably one of the highest priorities for us to understand, not necessarily to understand the exact ways that how the population moves, but to understand the variability, the uncertainties we have about movement in general, but also the variability from year to year. I hope that answers your questions.
Well, clearly it doesn't answer the full answer as one would like, but it's a very good explanation, and I can sort of try to get a feel for you know, how you would try and move on from here, because I think this is a critical point, and so thank you for the answer.
And I think that's something the MSE team is, one, hoping for feedback from the SRB on, is where do we move from here? What direction should we be going? But the MSE team on its own has already determined that investigating the variability in movement and understanding this operating model and the consequences of not only assuming movement in this way, but the consequences of different assumptions of movement are very high priority for us.
Alan, can I just follow up on that? I think you alluded before to a third explanation, which is that there are productivity differences among regions, right? So if 2A is higher productivity, then it wouldn't have to be a higher R0 or net immigration. So what kind of supporting evidence is there that there might be higher productivity, and how can you account for that?
Yeah, that's a really good observation, Olaf. And what we have looked at in the past is differences in the growth rates of the fish in these different regions and even regulatory areas. We've looked at historical ability for these different regions and even regulatory areas to support the catches and the population coming out of it. And of course, there's always that confounding between movement between the regions and actual, say, productivity, if you want to call productivity like growth and mortality in these different regions. But it's something that is a good avenue to continue investigating as well, is not only the movement between the regions, but when a fish is in a region, how does growth differ among the different regions? And then how does even natural mortality differ among those? So we need to work on disentangling those different concepts of productivity, I think, to help us understand those as well.
Yeah, I agree. And I mean, to me, that seems like a potentially productive research area, looking at spatial variation in growth and natural mortality, particularly since there's already been a lot of work done on trying to get empirical estimates of movement from tagging. And those just don't seem to match up. So trying to get at that question by doubling down on studying movement directly, to me seems like it might be less productive than trying to understand some of these other components that could give you the same pattern.
Yeah, and I mean, it'd be great if we knew that stuff. With the foresight of the MSE team, we have this operating model is very generalized and will accept different mortality rates, different weighted age, and other things to account for that productivity if we wish to. So, you know, maybe even the first route we go on this is to just look at sort of scenarios of what if mortality differs between these regions in this way? What if growth differs between regions in this way? And sort of investigate the consequences on management outcomes of that, so we can actually then begin to prioritize where this research should occur.
I think that is fantastic. That is, I think, exactly in line with a lot of conversations in the SRB. You know, if you can use that kind of scenario development to really prioritize the biological research, that's just the ideal kind of integration. And maybe you can do it in a way where you try to alter factors individually. How much spatial variation in growth would there need to be to make this fit? If you tried to balance it all with growth, how much would there need to be in terms of differences, spatial differences in mortality to make it work out? And you know, once you get, and I'm sure it's not one of these things individually, but once you get to a variable that, you know, with a plausible amount of spatial variation could account for some of these patterns, I think that's probably a good place to dig in on the biological side.
Yeah, and the MSE team is so excited to begin those types of investigations. And for now, you know, this year, 2020, has been our nose to the grindstone. We didn't even realize COVID was happening. We were all just so glued to our computer anyways. But we're so excited. We now have this framework to begin answering those questions. And I've been trying to, you know, let the commission know that we now have this framework, and it's much bigger than the MSE. It can begin to answer a lot of questions, as Dr. Branch was noting this morning in his review.
Yeah, that's right. I mean, they probably built this in order to test harvest control rules, but that's only a small fraction of what it can do. And I think, you know, in terms of guiding integration, that's a really valuable reason for having the MSE.
Yeah, thanks for that. Should I move on to talk about, oh, go ahead.
Well, yeah, I just wanted to add something. So I think what kind of stepping back a bit, you know, you've gone into the realm of spatial models, and we know from a lot of simple models of spatial population dynamics that the two main features that drive the sustainability of, say, a metapopulation are movement rates and the correlation in productivity, in the not necessarily the correlation in productivity variation. So, you know, when those two things kind of happen independently and can lead to different types of outcomes. And so those are really the two main drivers of a spatial model. And so what you're doing here, you're looking at the movement, assuming that, and you had said there's a coastwide spawning biomass. So basically that you're basically saying that all of the productivity variation is the same, and that all populations contribute equally to the total. So I think I would kind of step back. I have another comment on where to go, but just to step back and think about some of the main issues in spatial population dynamics, and those are two. And the issue is it raises so many, it can go in so many different directions, and it'd be important to control these directions because you have possible spatial variation in spawning biomass, and then spatial variation and annual variation in productivity, and all those sorts of things. And it creates a lot of dimensions, basically, is where I'm getting to. And so, somebody could say, well, you have to account for all this other stuff. Well, this has to go forward and produce answers on a certain timeline that's already scheduled, and then you're going to lose your team. Like, you don't know right now that you're going to get your team back. So I think it's important that we recognize that this current operating model is what was what you were aiming for 2 or 3 years ago, and now that you're here, you kind of have to get on with doing the management strategy evaluation work that was planned, and condition everything on this is the state of knowledge at the moment, right? This is the operating model state of knowledge, and not go too crazy on. Even though I'm a real advocate of multiple operating models, they have to be limited at the moment.
I would completely agree, Sean. And it might be a slightly different angle, but I see this as the start of something bigger. Now we have the framework, we have an operating model, we need to operate on this timeline, we need to produce results for January, which we are doing. And I think it is important, as the SRB in their SRB017 report stated, that it is conditional on the assumptions and the operating model that is currently created.
And I wouldn't, I wouldn't want to try to create the impression that some of these tweaks, you're going to find the right model, because that's not going to happen, right? That's not the purpose of MSE. So even, for example, showing low PDO, high PDO movement, I mean that's one possibility among others. So that's
So actually running a model with only one of those options in it, is that what you're saying?
Well, yeah, I would suggest that that would be a more complicated step, I think, doing that. And yeah, I mean, I think possibly a bigger issue is that having regional spawning biomasses, and then looking at the issue of correlation and recruitment variation. I think that's going to be a big deal.
Yeah.
And and then that's going to interact with the movement, right? Under low movement, it's going to have much higher consequences. Under really high movement, it's going to dampen it out. So, but anyway, a lot of this stuff is in the metapopulation type literature that the models are simple, but the messages are the same.
Yeah.
Even when you get up to these more, all you're adding here is an age dimension. So.
Just add one point here. The movement that you have here, or what you're making inferences on, there's going to be genetics data that are available shortly, and it would be really interesting to see how the genetics data, so you have movement probability. It's not NM in a population genetic context, but it would be really interesting to see what inferences can be made with the genetics data in terms of degree of difference between the populations, and sort of a back calculation of what migration rates might be with the genetics data to see if they stack up to what you're simulating here.
Yeah, I completely agree. And we're really excited about linking more with the biological research group.
Okay.
All right.
So we got that off our chest.
Yeah, good. So I want to talk a little bit about variability and uncertainty in the operating model, because again, I think that's the most important part of MSE to me is, evaluating management procedures so they're robust to the uncertainty and variability. So there's two types that we've been talking about with stakeholders and managers. And that's integrated uncertainty, which is really, just modifying the parameters and putting variability in the projections and then doing a lot of simulations so that we produce this cloud of trajectories under each management procedure. And that's what we mean by the integrated, is it's integrated in the simulations and just comes out naturally in the performance metric. We've been using the word scenarios, and this has been a long time, probably before I even came into the IPHC MSE, about scenarios, but these are those specific cases you want to investigate to investigate departures in an assumption, such as, I think what Sean might even mention there is, what if movement was always at the low PDO state? What does that look like? What does it look like if it's at the high? What does these results look like if weighted age is always at a low level? What does it look like if natural mortality is different among the regions in some way? Things that we're not sure are really have a lot of support for, but we're concerned about, and we want to potentially integrate them into the results, or at least show them on their own, so that we can say this management procedure might be more robust to that scenario than a different management procedure. So with that said, we've definitely integrated uncertainty into these results, but we haven't really gotten into the stage yet of investigating different scenarios. And this relates to an SRB request of the request about reducing or defining the scope of the MSE for this year, and potentially define to then display it in terms of a reference grid and a I can't remember the exact terminology that was used in the report, but a separate grid that is more, may not be in the reference grid, but more departing from the scenarios as more investigation. A robustness grid, I think, is what it was called. So we haven't actually gridded it out, but we've integrated the uncertainty just by adding in the variability specifically, and I'll show you a few trajectories that'll give you an idea of that. So what's being plotted here now is, again, the assessment cloud in the pink, and then in blue, and this figure is slightly different than the presentation you may have downloaded from the SRB017 website, because we had a lot more simulations, and I created this figure, I think, just 2 days ago, after doing a lot more simulations. And what this shows, though, are individual trajectories from the operating model, given the uncertainty and the variability. Well, this is all historical, so basically uncertainty in parameters, uncertainty in the movement parameters, and natural mortality, and some of the main parameters. And so it just shows you that, historically, it's just not able to get down to the low levels that the stock assessment got to. And I think this might be partly because we now have a multi-region model where it actually has to maintain biomass in all of the different regions, like region 2. Whereas the stock assessment model, it's operating on a coastwide biomass, where if there's mortality, high mortality coming out of region 2, it just can take that from a coastwide level. There's also, might be a little bit strict on our assumptions of parameters in the historical period, and we might be able to open that up a bit more to get to these lower levels. But what is comforting, at least to me, is that in the later few decades, we matched the assessment series at least really well, and we have uncertainty that is greater than the assessment is showing, even the ensemble assessment, where the pink is the ensemble assessment. So we're expanding the amount of variability in our projections from what the assessment thinks it may be. to show you the variability and the outcome on the predictions of stock distribution. I think that's doing well again, where now the cloud here, the shaded areas, the uncertainty in the operating model, given all the different parameters that we've put in to create these individual trajectories. And I think that covers a wide range. I was a little concerned in 4B that it was going down to 0% in its proportion, but it's not. I think the minimum might have been like 1% or something. So it's always maintaining biomass in region 4B, although it's just the scale, and I plotted these all to have the same scale. But again, even the uncertainty in the OM is not able to explain that 2019 data point. And I'm really looking forward to see what the 2020 stock distribution looks like, and if that is a continuing trend, something that we may want to be worried about. And then looking at the actual projections from 2019 onward, these are projections without fishing. And so I think there's 600 simulations here in the cloud of points, and I've only plotted potential trajectories from the first 20 simulations, where the dark line is the median of those. A couple of things that struck me. First of all, it seems to stabilize here, and then it goes to a new point and maybe stabilizes after about 60 years. That's pretty interesting. I have no idea why that's occurring. The other thing is that, you know, the changes in weighted age is driving a lot of these changes in the biomass trajectory, which does seem to match what has been seen historically in some ways. Remember, these projections are without fishing. There is a lot of variability without fishing, but when you are fishing under these mortality rate assumptions that our management procedures incorporate, the variability in the spawning biomass is going to be much less. So with that being said, we've had to make the decision, instead of simulating out 100 years for all of our management procedures like we did in this no fishing case, we've only simulated out 60 years. And this was just a matter of time. We didn't have time to do all the simulations we needed to do out to 100 years. Realizing that the more you tack onto this model, the slower it gets in its simulation, because it's doing estimation models and other things with a lot more data now. So we've cut that off at about this point in time. A little bit concerning if we're looking at no fishing, but the fact that we're looking at projections with fishing really squashes down the amount of variability, and I think it's going to be much more stable, at least give us a good indication of how these management procedures would perform 60 years from now. Something to think about. And then the final source of variability of concern is what we're calling implementation variability. And we've split this into 3 different concepts. These 3 concepts are all important concepts to the MSAB as well. The first one is that decision-making variability. And I think about these actually in chronological order. So we have, again, that management procedure, and it ends, and it's a procedure that can be codified, as Dr. Branch mentioned, and it ends with a very predictable outcome. And then we introduce implementation variability. First, the decision makers make a decision, which is the adopted TCE-wise may differ from the management procedure outcome TCE-wise. And this is one that might be hard to predict, but we do want to look more into it and figure out a possible way to simulate this. So now the decision makers have set a limit, and the fisheries go out to fish that limit, but they don't exactly catch the exact limit. Of course not. They get really close in our commercial fisheries, but recreational fisheries may be a little bit off, and then bycatch is definitely an unknown compared to the limits. So there's that, what we call actual fishing mortality variability. And then the third one is pretty interesting, and this one some people refer to as catch accounting. And even though the fisheries go out and the limit might be 10 million pounds and the fishery catches 9 million pounds, they have to estimate how much the fishery actually caught. And that's another level of uncertainty or variability. And the estimate might come out at 8.8 million pounds instead of the 9 million they actually caught. The MSAB is very interested in that uncertainty and catch accounting, or the estimated amount of, sorry, this should say mortality, not variability at the end of 3. But that uncertainty in the estimated amount of mortality is important because I think it differs among the different regulatory areas, and there is some talk of potentially accounting for the amount of uncertainty in setting catch limits, for example. So we realized that an SRB request was to look into the decision-making variability, and unfortunately, for us to just get the simulation framework together, we have not had a lot of time to look into the decision-making variability. So it's not implemented in any of these simulation results at this time. Potentially, we could add it in as a test case on a couple of specific management procedure projections within the next month or two, now that the simulation framework is together. And then I want to point to, when I first came on to IPHC in 2016, I did look at decision-making variability. It produced some radar plots in an early SRB meeting. That might have been like SRB 9 or 10. But I only had 3 years to work with because they just recently started looking at TCYs in 2013, I believe. So now we have a longer time series, and it would be useful to look at sort of the decision-making variability.
Sorry, Alan, on that, just on that previous slide very quickly. I think one of the things when I looked at some of these things in for one of our stocks for the commission here, it turned out that particularly in this case, it was the discarding rate or the misreporting rate, i.e. intentional misreporting that caused under 3, but I think the point was there was biases in these things. The behavior was going to be different when the stock was in a different state, i.e. when your quota in, or your TCOY in relation to the abundance is disproportionate, then you have a higher probability than in other cases. Have you contemplated, or have you looked at, or will you look at when you do these, re-look at the data on the decision making to see whether these stock dependent or periodicities are in there?
Yeah, for sure. And I guess it didn't come across in this slide that this could include biases. And I think those biases are a very important part of this process to include. So yeah, when we look into it, you know, depending how many years of observations we have, you know, for the decision making, for example, we won't have many years to look at, so we won't be able to see over a period of time when the stock was high and the stock was low. Just there's been so many changes in management and the management advice coming out of that. But for things like actual fishing mortality, we could have many years to look at and see how does the commercial fishery operate at high limits versus low limits, and it might operate very differently. It might not be able to reach those high limits, but does get really close when the limits are low. So those are definitely things that we will be looking into.
Thank you.
Alan, one thing to add to that list there is the amount of change from one year to the next in the harvest or the Fs. And that would allow you to go back even before the adoption of the harvest control rule. Because for many of the management strategies that imply dramatic shifts in harvest from one year to the next, those are the ones that are most likely to get hit with the decision-making uncertainty, right? Because the big changes in either direction are not likely to be followed if that's what the management rule is suggesting. And so looking historically at how much it's changed probably gives you some idea of what the management system's tolerance is for high year-to-year changes in the quota.
Yeah, that's a really good point. I hadn't thought about that, but thinking about it more on their tolerance level for changes in either harvest rates or even just general limit, absolute amounts might be useful to look at. Okay, excellent, thank you on both of those. So I just want to give a brief review of the management procedures we're looking at, and then we'll show some results. I think it's just useful to have this context on the management procedures. So again, remembering that a management procedure, we're thinking of it as the monitoring, the estimation model, and the harvest rule. What the MSAB has defined mainly is this last part. What are sort of these components of SPR and distribution methods? But we've also introduced monitoring and estimation models as part of the framework. And you'll see we have 3 different types of estimation models as well. So at MSAB 15, they identified in red, I won't read this slide, but just so you're aware, they identified a number of components or elements of interest, one of which included constraints on the changes in the TCY, and then, looking at minimums and defined shares between regions or regulatory areas, et cetera, and relative harvest rates. So these concepts were then turned into, we did an exercise at MSAB 14 with the MSAB, which I think worked really well. We gave them a sheet, a bunch of squares with that procedure that I showed earlier with coast-wide and distribution, and asked them to put elements in each of those squares that was important. And what came out of that was, I'll show you in a little bit, and I forgot my presentation already, but I wanted to talk about the monitoring first before I get into those specific elements. And that is, monitoring simulates the survey and fishery data in very normal ways that many different simulation studies do. So I'm not going to talk about that in particular detail, other than we've tried to mimic the variability on the indices, age compositions, and stock distribution as much as we could, noting that stock distribution is calculated from the generated indices, the generated data. And then for the estimation model, we have introduced, or we include 3 different types of estimation error. One, which I always think it's useful to look at with, is no estimation error. You basically know your stock management procedure without any input or general variability. And then you can stack on top of that estimation error. We've used simulated estimation error in a similar way we did with the coast-wide MSE, and that's basically adding deviation or variability to the relative spawning biomass, the stock status, and the total mortality that would come out of the procedure with a bivariate normal distribution. So this is just trying to mimic the stock assessment, the 2 outcomes from the stock assessment, and their correlation with each other. And we also have some autocorrelation on that as well. And then, so that's the assessment output, but there's also stock distribution used in these management procedures, and that comes from the variability generated from data. And then finally, we've actually included stock synthesis assessment model. It's been very challenging. With an ensemble, we're trying to include more than one model, and that's proven to be challenging. But at this point in time, we have one, the long coast-wide stock assessment model working satisfactorily. And that actually then predicts the spawning biomass, the total mortality from the stock assessment model, and then stock distribution from the generated data. So that stock assessment model actually uses the data generated in itself and does estimation. And I just want to give a quick shout out. Piera Carpi has done a fantastic job in integrating that into this framework. So that brings us to the management procedures for evaluation. The MSAB took all those different elements, as you can see here, and put them into basically 11 defined management procedures, where these 11 defined management procedures are, we could, as the MSE team, could go away and actually write them in code. And they also prioritize them, as they identified 6 as having a high priority, meaning put your focus on these first, give us high precision, meaning lots of simulations, and then the other 5, a little bit lower focus. We want to see them, but maybe don't have to do 2 billion simulations on them or whatever. So that's been helpful for us to prioritize a little bit our time. I don't want to show you all that table. It is available in the MSAB 15 report. And instead, we've put it into this table format, just showing the colors in the backgrounds here. Boy, I hope my kids aren't bothering you. If they are, I can tell them to go outside in the rain. But this table basically shows what each of these procedures with a letter has incorporated in its element. So it shows how you can compare procedures. For example, A and B are exactly the same, except one has the constraint of 15% on the annual change in the TCUI, and the other one doesn't. So you can look through here. Some important ones to look at are MPA is an unconstrained that mimics the current procedure, along with these two agreements for 2A and 2B. And the commission has agreements on the amount of TCUI in 2A and 2B, where 2A gets 1.65 and 2B gets a percentage determined by a specific formula. And then if we go over to G, we see sort of the past interim management procedure, which it does have a constraint on it though, but just using 032 stock distribution and relative harvest rates for 2 to 3A and then 3B to 4, where 3B to 4 is harvested at a lesser rate. And that one G does not have these agreements. So we can really see the effect of the agreements in management procedure G. And then we can look at different relative harvest rates, for example, in H, where we have only a smaller relative harvest rate in 4B, for example. So those are the management procedures, and reminding you that there's 3 assumptions about estimation error. I'd like to switch over to the MSE Explorer and show you a few things from that if you're okay to move into that realm.
I just wanted to add one thing.
Okay.
You know, it's not critical at the moment, but for the stock assessment model 1, the data that you're simulating for the assessment, is that age structured data?
Yes, it is age structured data, and then we're using age and length keys to classify O26 and U26 concepts.
Okay, so this is this is one of those integration things where you could also point out the need for the logistic normal in this context as well. I don't know how you're simulating that age composition, but if it's from a multinomial, it's going to be pretty well behaved.
Yes. Well,
so it's probably going to be unrealistically good, whereas, you know, further research on that logistic normal and working out at least some basic correlation ideas would create more realistic data for that simulation.
Yeah, so we are using the Dirichlet to generate the age composition data, which does allow for some correlation, but it has its issues, as has been pointed out by Ian as well as others. And so we've had to make a few assumptions about when sample sizes are small. You know, it doesn't quite behave very well, for example. But I think, you know, as we investigate the logistic normal more, it would be useful to bring that into this as well.
Okay.
Anything else before I move to the Explorer and show you a bit of results? So this website is live. And actually, just so you're aware, I archive what I show at each meeting. So there is another website that is for SRB017 as well. If we ever need to go back and look at what was seen, for example, at MSAB 13, that is available online as well. And I've actually looked at that recently. So that's all available on this website like this. And zoom in a little bit and just give you a brief overview and then show you how it's used and then show you some outputs of this first. Up here on the top, and stop me with any questions at any time, but this is basically what the MSAB will use to do their evaluations. A lot of times I drive it, but MSAB members might actually be using this website right now. So if it starts to fail, I might switch to something a little more reliable. So up here, we have different pages that you can view, a table of plots, trade-offs. You can click on MPs to get a reminder of what each of these management procedures and what they incorporate, for example. there's help, which we definitely need to make a little bit more, as Dr. Branch mentioned this morning. It'd be nice to have more guidance on how to use this tool. In the description, we usually have a grid down here which shows what's available and what's selected. That I need to do some fixes on that, so it's not available at this time. We've supplied updates so that everybody's aware when it gets updated. So if you want to look at this at a later time, have a look there. The first thing to do when you come to this page is either refresh or press F5, because it doesn't always load the most recent version, for example. Down here on the left are the different options you can select for the management procedure. So these are those estimation error types, not, as part of the management procedure, but not really a decision to be made in choosing a harvest rule. But then we've had constant total mortality options in the past. I don't think there are none available right now. And we have 3 different SPR values available here, and that will be expanded as more simulations come in. And then here are the specifications for the management procedure type, basically the distribution components being A through K. Down here, even lower, are the different time periods that have been defined by the MSAB. The long term right now is at 50 to 60 years, as I noted earlier. And then you can choose the different regions. Some performance metrics are by region, and some performance metrics are by regulatory areas. What comes up first, I'll show you the table. You can see the different management procedures across the columns. And then down here across the rows are the different performance metrics. What appears first are the performance metrics that are associated with the primary objectives. And what the MSE team is currently working on is a page that actually really specifically links the objectives, lists the objectives, and links the performance metrics so that you have a really clear view how to evaluate this against the objectives. And that's something that we're working on at this point in time. But for now, many members know how these are linked to the objectives. So what comes up first are the primary performance metrics. And you can see there's none by regulatory area because we haven't selected regulatory areas. As soon as you select a regulatory area, new ones pop up, such as the TCY in area 2A. So I'll just delete that for now and get back to this coast-wide performance metrics. So the first thing that I wanted to show you in this table was to do a quick comparison between the different estimation error types. This is really important, partly because in the coast-wide MSE, we found that the variability from year to year in the TCY was quite high. And that was a big concern to the MSAB members, at least, and that's why they developed management procedures with constraints. So what I'm going to do is I'm going to back off the specifications into only management procedure A, which is the only one that does not have an annual constraint on it. So we can really begin to see the variability and the differences between them. We have no estimation error, simulated estimation error, and then the stock synthesis model, that single long coastwide. And what you can see, and this is all for an SPR, input SPR of 43%. So we have the probability of the spawning biomass going below 20%, the probability it goes less than 36%, and then some of the TCY values. So there's definite differences in here. The important ones are these stability ones to me right now, and that's showing an AAV in the TCY of about 4.4% without estimation error, and then up to 6% with estimation error. And there's a slight difference between the stock assessment model and the simulating estimation error. There's a bigger difference in the probability that any 3 years have a TCY changing by more than 15%. And you see that's a probability of 13 or 0.13 with simulated estimation error, but a much lower probability for the stock assessment model. So there's some other performance metrics that can give us some insight. So I'm going to turn on the median average SPR. So this would be the actual SPR realized from the application of the management procedure. And then I'm going to turn on median average RSB, which is stock status or relative spawning biomass. So now to click those on, they appear up in the table here. And the first thing to notice, so the SPR coming out of the simulations with the input of 43% and no estimation error is 43%. So, whew, good thing. They seem to work. This might not be the case though as the population gets lower because of the control rule interacting with that. So the control rule actually changes the SPR to determine the catches. And so it's a fun metric to look at higher fishing intensities, for example. Now, when we're estimating simulation error, that comes up at about 43% as well. So that's unbiased, basically, determination of the fishing intensity when we're estimating simulation error. What's different here is with the stock assessment model, you see that the SPR is slightly higher than your input SPR, and that's indicating there's some bias in this long coast-wide stock assessment model. It's a slight increased bias, which we actually show in the MSAB's 16 document that was recently published, that generally the long coastwide has a slightly lower fishing intensity than this operating model would simulate it and the data generation would produce. And of course, then that translates into different stock statuses on average, where we're about 37% for simulated and then 40% because our actual fishing intensity is not as great, or the SPR is actually higher for that case. So where we're at with sort of where the MSE team is at with this is we've really been trying hard to bring in the assessment model, because that's really what we want to do. We want to bring in those biases. We want to bring in the autocorrelation that the assessment model sees by entering in data year after year. But at this point in time, we're not certain that we're mimicking the ensemble assessment very well. We're seeing this bias. Initial investigations with the short coastwide estimation model, or the stock synthesis model, has indicated that the bias is the opposite, and that the ensemble, there's a paper that Ian Stewart was lead author on a year or two ago, the ensemble actually stabilizes the output than just compared to a single assessment model over time. So we're a little bit concerned that just using a single stock synthesis model may not be characterizing the stock assessment correctly. And at this point in time, we've focused more on simulating estimation error. It's similar to what we've done in the past with the coastwide, and it's a stepping stone moving us towards getting the ensemble to simulate the ensemble in a more realistic manner. So I think that's useful for the SRB to think about if they have any opinion on, should we just be jumping right up to a stock synthesis type model, even though we only are able to use one right now. And I suppose I should explain why we're only using one, is because we just can't get good behavior out of that short coastwide assessment model at this point in time. Pierre's been working really hard on that. She's made huge gains on it. There's just a few more concerns that we have, and so we've switched over to focusing on the simulated error with some comparisons to the stock assessment model at this point in time. So with that being said, I want to then eliminate the, that's interesting. I'm learning things already. I'm going to bring in some of the procedures. These are the priority procedures with simulated estimation error and also the other SPR values. So actually, yeah, so you see that this gets quite big. Let me take this out for a second. So you see that it grows really quickly as you start adding all these different elements. And this is going to be the challenge of the MSAB, and we'll be helping them along with this, as well as our own evaluation. And then the commission is distilling this into a small set of manageable results. But one way to do that is, instead of showing it in a table form, is to show it with the plots. And I haven't shown you the plot page, but it basically displays all the performance metrics that are currently selected for the various management procedures in a way that's hopefully helpful. This is basically the number of simulations that were done, so people can see, like H doesn't have a lot of simulations yet, so it might not be as precise as others. I'm going to turn that one off. And this will just focus on the main performance metrics coming out of this. So we have at the top, the different colors indicate basically different SPR values. We need to improve that labeling a little bit. So we have blue as a higher fishing intensity, which shows that it has a higher probability of going less than 36% of B0. So that's good. And then red is the lowest fishing intensity. We can look at, say, the median average TCY and then the AAV, how that changes across the different management procedures. Now the management procedures are along the x-axis. So a couple of things first. The main biological objective here is to look at the relative spawning biomass and its probability of going less than 20%. And what you can see across all management procedures, it's much lower than the 10% threshold defined in the objective. And it's basically 0 for all of these, which is a bit lower than it was for the coast-wide MSE. So it's showing no probability at these SPR values of dropping below 20% B0. Remembering that the control rule is acting here. If the stock status is less than 30%, the control rule kicks in and it's not going to want it to go below 20%. And the other thing to remember about this is investigations of what potential relative spawning biomass associated with fishing at MSY would be are down around 30% in some of the investigations we've done recently. So operating at SPRs of 40 to 46% are much higher than MSY, and we don't expect there to be high probabilities of going below 20%. But going less than 36% is definitely changing across the SPRs. The TCY is changing across the SPRs, although the range here is only between about 49 and 55 million pounds, so not a huge change. The constraint, which A is the only one without a constraint on the annual change in the TCY, has a big effect on the stability. And so you can see even the AAV is now very high when fishing at a higher fishing intensity because the control rule is probably being enacted more often as well. But that constraint really dampens that down. And basically it shows there's 0 probability that the TCY is greater than 15% in any of the 3 years, which I hope that's pretty much the case because that's the constraint on the TCY. So that's some results on the coast-wide level. So let me just turn something off here real quick. Turn off it being less than 20%. And I'm going to turn off the change in any 3 years because I want to start looking at some regulatory area ones. And I'm going to turn off the probability less than 36% as well. And so now we have basically the TCY and the AAV on the TCY. And I'm going to look at regulatory area 2A. So what we have here, remembering A is without a constraint, and then all the others have the constraint, but A and B are the only 2 management procedures with an agreement for 2A and 2B on getting a defined percentage for 2B or getting a defined amount of 1.65 for 2A unless it goes below that for some reason. And so now what you see, we have area-specific performance metrics such as the median average TCY in 2A, the minimum TCY in 2A, and then the percentage in 2A. And as expected, A and B are much higher than the other management procedures, and there's some difference in the other management procedures. So I don't want to get into specifics of the results. I'm just trying to show you a general way that we would be working with MSAB and the commission and analyzing these different management procedures. And also realizing that it's going to get very complicated as we add more regulatory areas. So for example, let's add 2B. And now what we see is if we look at, so for example, the median minimum TCY in 2A and the minimum in 2B, is we have different trends. So what F has is a national share for 2B, and it takes away the agreement for 2A. So there's the trade-off. 2B gets more, 2A gets less. And so what we're going to have to do is really begin to focus on the trade-offs between these management procedures and the trade-offs between between regulatory areas. Because we can't just simply use statistics now to find an optimal. We could, but there might be other objectives that aren't in these primary objectives that they want to manage them on. And I think it's really useful for us to realize the trade-offs. And we'll do what we can with the performance metrics and finding an optimum solution. But it's going to be a challenge with 8 different regulatory areas. So we're going to be showing them results like this. We're working on new ways to present the results. We're very open to ideas from the SRB on how to present these results. And as the MSAB reviews this before MSAB 16, we're asking them for any comments and plots and other things that they might find useful. So I guess the MSE now is in simulation mode, it's in production mode. We're filling out this table, and as we do those simulations, we're beginning to focus on how to present the results and give MSAB and the commissioners the information that they need to make the decisions they need to make. With that, I mean, we could turn on all sorts of things, but I think you get the picture of how this works. There's results here. If you want me to show you anything, I can. But that's basically how we're going to present the results. And if you want to ask questions on that now, otherwise I think I have 2 or 3 more slides to present just on the program of work and what's expected throughout 2020.
Thanks, Alan. Yeah, that's quite a bit of progress, and pretty impressive. I love that table, that design table on slide 29. That's great.
It's helpful to get the big picture, I think.
Yeah. To get people involved in seeing like all the choices that they have to make and how some can be simple and some can be hideously complex, and whether those 2 may give you the same answer. I was wondering if you have like pairs plots, just for regulatory area by regulatory area, pairs plots of catch or something.
Yeah, that is a really useful. What we're working on, see the trade-offs. We haven't really worked on updating the trade-offs tab yet, but this is something where we need to spend some time. I think actually, let me see if this will work.
Because it would be really good to see. Oh, yeah, cool.
So this is the TCY and 2B versus 2A, and the problem is I think, I don't really know what's going on, but I think this might, no, oh no, it's not splitting it out. So we need to work on ways to, there we go. This is by SPR now. So we can see, but we need to make it work for different management procedures, which I guess it's not working right now.
Yeah, I think for a particular procedure,
to
look at I mean, you could, I guess, look at the median, TCEY across them, just to give an idea of, like you were just pointing out, if 2B gets what it wants, then who's going to knock at what they want, and it may not just be the adjacent ones, it could be.
Yeah, that's a good way to put it.
Definitely more distant.
Yeah. Yeah, I guess if we plot something like A versus F, it didn't quite work out the way I expected it, but yeah, so
that's fine. That's fine. I just want to.
Yep.
Let's see, you're on it anyway.
Alan.
Yes.
Just quickly, I do like. Well, they're not what I'm interested in, but that's not the purpose. It's not for evaluating this. But I do like the way that some of the things can be compared, and I was playing with it the last couple of days. I think it's that table that you showed, or the matrix that that I was really lacking in order to be able to make sense of it. Yes, that one. And maybe there's a way that you can superimpose that when somebody hovers over the legend or something. I don't know. That's not important. But I thought it was valuable to say that really when you're doing those plots, you're connecting the lines, and really they are bar graphs rather than lines, because the x-axes are independent, or there's no order to them.
Yeah, yeah.
And I find it, stupid. I should be able to read it, but I find it very hard to actually read those graphs when they're in that form, because there's always the implication that there is a gradient along the bottom, as there is in those other plots that you showed. So I think it's important to make that distinction.
Yeah, and I think if we did, yeah, you're right. It's
for those it is okay. Yeah. For the other ones, it isn't really okay to have them as lines when you're comparing the metrics.
Yeah, no, exactly right. And what we're working on is actually having a tab up here to plot SPR along the x-axis. And in that case, I think we can have a line. People may want to interpolate between different fishing intensities.
Yes. Well, if you order that, you can say, well, in the highest fishing intensity, that's, and you can go down and you can figure out which one that is. But right. When they're unordered, you need to try and turn it into bar graphs. Sorry.
No, I get it completely. Yeah.
Okay.
Would it be useful to just finish up the presentation and then we can go into discussion? Okay, great. So just the last couple of slides is just a reminder of the program of work. This is something we'll be discussing with the commission, beyond 2021 as something we'll be discussing with the commission more, noting that, I'm really advertising this framework as something that we can use for many, many years to investigate a lot of different questions that the commission may have. But looking more specifically at 2020 leading up to the annual meeting in 2021, at the bottom of this table, we've been through the MSAB meeting and the June SRB meeting. We had in August an MSAB special session to examine preliminary results. And unfortunately, the fishing intensity wasn't working as we had hoped at that point in time, but we were able to show the MSAB some preliminary results of a single fishing intensity to get them used to evaluating the differences between distribution procedures. And it was a great sort of dress rehearsal is what we were calling it for what they will be doing in October at MSAB 16, is reminding them what the performance metrics are, what the objectives are, what they have to do, where they go to get all this stuff. So instead of spending a day doing that MSAB 16, hopefully we've sort of nipped that in the bud at this point in time. So here we are at the SRB meeting reviewing really penultimate results and the framework in general. And I've noted that's on schedule and results are being populated as we speak. And then at MSAB 16 are reviewing those final results with the MSAB putting forth their report to the commission on any management procedures and evaluations that they do. And then the annual meeting, with the interim meeting before then is presentation of this first complete MSE product to the commission for them to develop recommendations on a management procedure. So it's been a very busy year, but it's been a very fruitful year, and we're super excited now that things have finally come together. A few stressful points, and then as Ian did yesterday, he said it's nice to show a few pictures from the field, and I thought I would do the same. This is from the field from the perspective of a MSE team member, and so on the upper left we have what the computer might look like on any given day and any given location actually. This was actually taken in Canada, and that's my computer that I'm on here today in Seattle. So I never travel without my desktop computer, even nowadays. During the COVID era, you always got to figure out things for your kids to do while you're working. So I just give them a camera and they take pictures of me. And then this is a MSE team meeting with social distancing and the distractions that may occur when you're trying to have those meetings outside. Doing the safety the proper safety measures when looking for halibut in the Seattle area, which we did find one, as you see in the background there. Training to, the weight that we have on our shoulders is pretty big these days, so we have to train for that, as you can see here. At least we have one quantitative science member that can carry a lot of weight on his shoulders. And then we're always looking beyond our world for anything of interest, you might say. And this is a picture by Dr. Webster that he took just a few days ago. And so that's pretty interesting. We haven't found any halibut out there on this planet yet, though. So just a few observations from our field work this year, just to bring light to that. And then I provide a couple of recommendations here that are in the document. These are really generalized. And after the MSE team had discussions about the current state of the framework, we have provided the SRB with a few suggestions where we would like to get some recommendations. And you'll find that in the report that Dave has emailed out to you this morning. So with that, I will leave that slide up and we can have any questions, discussion as needed.
Speaking of things that happened during COVID, did you guys get to see Comet Neowise?
That was thanks to Dr. Webster again. He is the one who reminded us of that and took an amazing picture of it. That was really cool. Yeah. So I think Ray's online if he wants to tell anybody anything, but he's really into that.
Maybe we can give him an assignment for tomorrow, which is to make a slide.
Yeah, that would be great.
I saw it from far away through binoculars. I didn't get a good picture of it.
Yeah, me too.
I don't have to wait 7,000 years to see it again. Anne-Marie has a suggestion. So
well, I think that was
is that popping up on your computer?
Yeah, that was an email to me. I forgot to turn off my email.
Okay.
Yeah, she's really good about that. She should be part of the MSE team, as Carrie should too. They've had a lot of great suggestions.
Great. Okay. Before we get to questions, I just wanted to ask one clarification here. So, we were asked to endorse the FIST designs.
Mm-hmm.
In particular, what we're going to suggest is that we don't necessarily want to be endorsing designs because they may not be the ones that are actually implemented as new data roll in. What we're interested in is endorsing the process that's used to create the designs. So I'm wondering about B here, whether is it a recommend that we're asked to do, or are we going to endorse? This is a fairly significant step, I think, for the commission that they're undertaking this year. They're going to recommend a management procedure.
Yes, and the way that I'm seeing this, if I may speak to this question, the way that I see this is the MSE team, and the commission may see it slightly differently, but we would just like the SRB to, given what you've learned about the framework that we've developed and the review that Trevor Branch has given, that you recommend this as a MSE framework that can be used to evaluate the management procedures. One, that there's the operating model as is currently stated, and we've put in some potential language into the report for you to consider. But I don't see, actually, I did think about the word endorse, and I didn't see it as really the right word at this point in time, but more as a recommending that we are on the process of a proper MSE, and we have a simulation framework that can be used, and we have an operating model that's conditioned and can be used to provide advice on management procedures, conditional on the assumptions and other things in that. That's just the way I'm seeing it.
Okay. Speaking of the review, I just want to add something about exceptional circumstances, which I think is an important point to raise. I think we might have raised it at some point in the past, but I think what Trevor was describing in his review is not necessarily exceptional circumstances. It's the way you decide whether the advice you're getting from the MP is reliable or not, is whether the information that it's using is credible or not. So typically the way this is done is that you would make a projection under the operating model of the types of data that you would see. Let's say you're making, you're deciding on a 3-year quota. Then you would make a projection over those 3 years of let's say the FIS index in each region. And if any of the real data fall outside of that simulated distribution, then that's the kind of thing where you might invoke exceptional circumstances to decide what to do, because now the data that's being used in the procedure is falling outside the range for which it was conditioned. So I don't know if you have time to do that or not, but that's something you really have to think about doing, because kind of the first thing that'll happen, right, is, and we had this happen to us with sablefish. We set up a procedure, everything was great, everybody shook hands, off we go, and like the second year of implementing it, it was like, whoa, the survey index is way outside what we thought, and what are we going to do? So
Yeah, I would agree with that. And I think the bluefin tuna process, that's how it was explained to me. They saw something occurring with the population or the catches or something or the survey that was way outside their projections. And so they've invoked an exceptional circumstance to then bring back in the MSE and make any fixes as necessary.
Yeah,
so,
well, for southern bluefin?
I thought so. I
yeah.
Thought
Well, I just went through this, and it's never actually been used.
Okay.
It looked like it was going to be used, but so basically led to a bit more digging and found the error in the data, or the way the standardization model was treating the data, and it was fixed up that way. But
yeah, and so at IPHC, would this be something that we sort of describe at this point? And describe, as Trevor was saying, this is something you want to predefine. So we would predefine some things that we may want to look at over the next 3 years to make sure that we're still on track.
Yeah. So like the graphs you were showing before about the proportional distribution and biomass among the regions. And you noted that in a couple, I think in the most recent observation, it departed, like there's a different trend between the model and the data. Just pop up to that. Yeah, that R3, actually a couple of these, R2 and R3. So if you continue along here, and like these would be, borderline exceptional circumstances, right? A little bit, if these trends continue, and now that observed data, I guess it's model survey data, jumps outside of that region, that uncertainty region, then you're dealing with data that's outside of the realm of possibility of the operating model. So at that point, you would want to have some rules in place to decide, okay, how far does it need to be outside of that range? And if it does fall outside of that range, let's say it's falling, on the high side or on the low side, what do you do? It's weird. Like, it's a weird thing, because it's almost like you're creating another set of rules, but you're not going to test these rules. You know what I mean?
Yeah, for sure.
So you don't want to go too crazy. I think the point is for us, for what we do for Sablefish, for instance, we look at it and decide, okay, it's time to revisit the operating model. Let the procedure go. But if we hadn't planned on revisiting the operating model for 3 years, and now in the first year out it's departing, then we just move it and redo it.
Yeah, and I've also heard Doug Butterworth talk a bit about, that the MSE process should be repeated, or at least, investigating an operating model every 5th year or something like that, that it's on some time schedule where you're getting new data in and you're just doing an update and making sure that you're still on track, or, an exceptional circumstance might cause that to happen before then. Do you think that's worth it here?
Yeah.
I say that we're still in an infancy of developing, the full range of variability that we should be simulating in the operating model. And I think there's a lot of work that we should still be doing. We might want to be on a bit more frequent look at the operating model, at least in the MSE.
Or you just have a plan. Like, you have a plan where you look at these things. I don't know what the plan is. For in some cases, we typically choose a procedure and say, okay, it's going to work for 3 years. We're not going to really question it unless one of these exceptional circumstances changes the game, right? So, like for CCSBT, they used the Bali procedure for 10 or 11 years without ever changing it. And then, there were new data that came available, and that's when close skin market capture came out and everything. So it was all redone, and now there's a new procedure, and it's kind of open-ended as to when that thing is going to be redone. In that case, but yours, you might have a set plan. You might say every 3 years, every 5 years.
Yeah, I don't know if that's something that SRB might want to consider, is recommending the commission at this state of the MSE how often that might be done.
Yeah, I think it's too early to say that, but I think it is something where, in terms of developing exceptional circumstances rules, that maybe that's what it is. It's you can't really say what the frequency will be now, but certainly if you encounter exceptional circumstances, you should definitely redo it.
Yeah, I think that's great advice.
Alan, just a quick question that sort of relates to some of that, in the sense that Trevor, I think, made the point that, this will have to run along the current procedure for a couple of years before you can use it, kind of thing. And so I guess that potentially gives you a little bit of time if that's what, is accepted. But how does that relate to your comment or your recommendation that it be used for evaluation? Because surely he doesn't trust it enough to use it in management quite yet. And at the same time, you're making a recommendation that it be used to decide on management. Do I misunderstand that?
I haven't read Trevor's report yet. I haven't actually seen it, and so I'm not sure that.
Oh, sorry.
He was saying that. But I think, one, the commission is expecting results to look at and to potentially make decisions on. And so we've put the results from this framework together this year in the best possible way that we could, that would be useful for the commission to begin to understand the differences between the management procedures that are of interest to them, and that's these different distribution procedures. Now, and so that, I think that's where I'm leading to, and the recommendation is, and the SRB in their 16 report also stated, that results are conditional on the assumptions going in. I can't remember the exact wording, But, you know, is along that way is that define the scope of your MSE to be what you need for this upcoming year, and then just realize that the results from that MSE are conditional on that scope that you have defined. And so what we have here is a relatively narrow scope. There's a lot of room for improvement, I think. There's some things that need to be implemented, such as decision-making variability. But given what we have, you know, there's useful information here to begin the discussions on the appropriate distribution procedure for the Pacific halibut. That's at least my opinion of the work that's been done and the results that we'll be presenting at the annual meeting at this point in time.
Yes, and I think that clarifies it. Thank you. But I think we'll just, if we go down that road, then we'll have to be a little bit more specific than what you provided as to, you know, what bit you can use it to evaluate and what the caveats are. So, but yes, thank you.
Yeah, thank you.
Any other comments at this point? Okay, hearing none. I believe that takes us to the end of this session and some drafting for the SRB. So in terms of tomorrow, as I mentioned right after lunch there, I would like to use the entire 9 to 11 a.m. time slot for drafting the SRB report and plan on a hard stop for us at 11 so that we can review the report from 11 to 12. Any objections? Changes? Okay, well, with that, I think somebody's speaking.
Sean, if I could just chime in. I'm about to send you a version 3, which includes some of the summary points from the discussion you've just had on the MSE. So I'll send that to the SRB for your next drafting session. Thanks.
Great, thank you. And I also got a note from Keith that there was a separate site set up for us, a separate go-to-meeting room.
Yes, that's correct. So we'll close out the meeting now. We'll all log off, and then if the SRB just goes ahead and logs into that, you can have a private drafting session.
Okay, so for the SRB members, you have that in your email, an email from Keith from 10:12 my time anyway, West Coast time, 10:12 a.m. email that you can just hit that link and we'll reconvene. Maybe in 5 minutes. We'll take 5 minutes to take a break and then reconvene. So thank you, everyone. Great day of presentations, and we'll see you tomorrow.
Thank you, everyone.
Thank you.